Okay, today we're going to talk about the drug safety in clinical trials. So before I get into it, I think safety is a huge topic. In drug development, starting at very early stage, we do animal toxicity, and we want to look at drug safety. So in other words, without understanding how the drug does in animal, it is not ethical to progress the drug into human testing. So there's a preclinical safety findings, and of course, at preclinical stage, they had to identify this thing they called NOAEL, which is non-observed adverse event level. And that NOAEL get translated into human, and then we get into clinical development. So this preclinical safety, there's a lot of thinking and consideration in preclinical safety before the drug moves into clinical. And then, of course, we know in clinical development, we have phase 1, 2, 3 clinical trial developments, and we have to watch safety very carefully, very closely. And if the drug is efficacious and safe, get approved by regular agencies, and then it get on the market. So the post-marketing safety surveillance is a huge topic. So if you look at safety from in a drug development process from 3 stages of time point of view, there's preclinical toxicology studies, there's clinical trials, phase 1, 2, 3, and there's post-marketing stage. What I want to focus today will be just on the clinical trial part. Actually, the clinical trial part of the safety is sort of it's doable, it's manageable, because we know every patient who was recruited, we know all the events we can report and things like that. The more difficult challenge would be the post-marketing after drug is approved on the market, because post-marketing, I think the biggest problem is about denominator. So after the drug is approved, and within the next maybe a few months or half a year or something, you started hearing about adverse events reports and things like that. And what you can get is number of events or type of events and things like that, but but you do not know the denominator, right? Denominator in terms of how many patients at what dose and exposed for how long and that kind of stuff. So that's the most difficult part. We will talk about denominator. Denominator is well, in general, we call it drug exposure. It's looked at from 3 different dimensions. One dimension being number of patients, the next dimension being dosage, and the third dimension being duration. So it's how much drug being exposed to how many people for how long. So that part within clinical trial, you have all the data. So we have no problem finding denominators. So I want to just focus on today's talk will be on the clinical development part, phase 1, 2, 3. In clinical trials, we had to have a clear understanding of the difference between efficacy and safety. There are 3 major differences between efficacy and safety. Efficacy is a confirmatory practice. In other words, you sort of you want to test hypothesis to make sure that the test drug works compared to placebo. Basically, from hypothesis testing point of view, you have 2 distributions. You have a placebo distribution, you have a test drug distribution, and the hypothesis is trying to differentiate these 2 distributions. So by increasing sample size, we can sort of separate these 2 distributions with the pre-specified alpha and beta, we can test this null hypothesis. Safety is not. Safety is exploratory in nature, because at the design stage, you would assume the drug is safe. All right. If you cannot assume the drug is safe at design stage, it's not ethical to run a trial. The reason that we run the trial is that we believe the drug is safe. So you're not going to separate these two distributions. You know, especially when we're comparing test drug and placebo, you want the test drug safety profile will be as similar as the safety profile from placebo as much as possible. All right, so you don't want to separate the two distributions for sure. But you're learning from the safety data. All right, so that gets to the next point about observe versus design. Efficacy is a design feature, and safety is an observed feature. You design the study trying to separate the two distributions, you try to test hypothesis, it's all about efficacy. But because you're collecting data from from the participants who entered in the trials, so you have to collect their safety data, and you try to observe what are the potential safety concerns if the drug is being dosed for a certain period of time. The third difference is that efficacy is a population phenomenon, and safety is an individual phenomenon. In other words, if you want to prove efficacy, you have to have two populations, and you can separate the two distributions. So there are two distributions. You separate them, and then you claim not that the drug works, but you claim the probability the drug does not work being controlled under alpha. So you have to look at these two populations. In other words, each individual patient in these two distributions has a probability of 0. These are, so if we're looking at no more approximation, or we look at continuous data or whatever, you're looking at that bell-shaped curve, and the probability under that curve can only be calculated in the range. So for any point under that normal distribution, it has a probability of 0. So from efficacy point of view, you're not looking at each individual patient's change from baseline value. I mean, that's not meaningful. Or from an efficacy analysis point of view, you're looking at the mean differences. You try to separate the two distributions. Safety is different. Safety, if there's a very severe safety event in the trial, you may have to stop developing the entire drug. All right, so every individual event, individual patient in safety analysis is important. So this is the nature of the two differences of the three differences between safety and efficacy. So safety and efficacy, we have to look at it in totally different point of view. From this understanding, we know p-value makes no sense in safety analysis. And if you go to different teams, different companies, there's always argument about putting p-value for safety analysis or not. And when people try to argue to have a p-value in safety analysis, is that we want to use a p-value to serve as a flag. So if we see a difference between the test drug and the control with a small p-value, and then that'll give us a warning signal that we have to pay attention to this particular event. And I say that's not necessary, because all we need to know is to look at the percent differences. I mean, if the differences between test drug and control in percentages, like exceeding 3%, 5%, or 10%, that's strong enough a flag. You don't need a p-value. The reason I'm strongly against p-value in safety analysis is that you cannot interpret that p-value. a p-value is only meaningful when it's compared to an alpha. So from a design experiment point of view, when we design the study, you calculate the sample size based on your alpha and beta and everything like that. So you have an alpha to compare with. So for safety analysis, if you see a p-value of 0.02, I cannot interpret that because I don't know whether 0.02 is being compared to 0.05, 0.025, or 0.0125. I cannot interpret that value. Or I don't know the alpha. And of course, in safety analysis, there's so many events, there are so many comparisons. You have to make a model comparison adjustment, and all those kind of questions are unnecessary. Just don't provide the p-value. So today we're going to start with ICH guidance. I talked about ICH before. Actually, this organization has made a lot of important contributions to in standardization of drug development and drug regulatory. So we'll start with ICH guidance, and then we'll get into safety evaluation. The first bullet point about exposure study discontinuation is about denominator. So from clinical trial, the denominator is there. You have it on hand. It's in your database. And then we're going to talk about adverse events. And after adverse events, we're going to talk about clinical lab and vital signs ECG. The basic difference between AE, adverse events, and all the other safety findings is that AE is subjective. It's from each individual patient and from the physician's observation. So adverse events has to be sort of something reportable or observable, or reportable from the patient. The patient has to feel possible AE so that they can report, and then the physician will have to be able to observe, and then that's an AE. But lab, vital signs, and ECG, these are what we call objective findings. These data are collected from some kind of equipment or device. They're not a subjective feeling or or something you can observe and say your red blood cell is too high, your white blood cell is too low. You cannot observe that. All right, you have to draw the blood. You have to go through that lab evaluations in order to understand whether there's abnormal lab value or not. Vital signs, the same thing: blood pressure, temperature, pulse rate, these kind of things. You have to measure it. ECG is another kind of objective findings. We sort of talk about ICH E14 when we talk about phase 1 studies, and that's about the QT prolongation, these kind of issues. And you can study ECG from the ECG machine or Holter monitors. So after these major components of safety, we're going to talk about integrated safety analysis, ISS. And then the PSUR, periodic safety update reports, are post-marketing observations or findings. So we're not going to pay too much attention on that, but today we're just going to focus on within the clinical development stage, how we handle safety data. So ICH, actually in 1990, there was this International Conference on Harmonization, and then like I said, they made a lot of contributions, and they developed guidelines. They set up CTD, and they came up with MADRA. Later, we're going to talk about MADRA. In 2015, so after 25 years, they decided to change it to International Council on Harmonization. So we're still talking about ICH, but that C has a different meaning now. It's International Council on Harmonization. So ICH, to begin with, was not a sort of official organization, but over time, well, even though they're still not official, but they become a very organized, very well organized uh, association, and they're making important contributions. And right now, there's many members. The members are from different countries, different legal agencies. So legal agencies and research-based pharmaceuticals from the different regions, they got together, they came out with the guidelines. All right. Objective was to increase international harmonization of technical requirements to ensure that safe, effective, and high-quality medicines are developed and registered in the most efficient, cost-effective way. The guidelines represent the state-of-the-art, agreed upon scientific guidance for meeting technical requirements for registration. E1. So there are sort of three major categories of guidance in ICH. ICH-Q is about CMC, chemistry, manufacturing, and control. ICH-S is about ICH safety, is about preclinical. And ICH-E means efficacy, is about clinical trials. But ICH-E1 and E2 are still talking about clinical safety. All right. So E1, the extent of population exposure to assess clinical safety for drugs intended for long-term treatment of non-life-threatening conditions. So the first thing is to look at denominator. All right. So in safety analysis, the most important part is to find your denominator. So number of patients and duration and of exposure and also dosage. Then we got E2A about clinical data safety, clinical safety data management, about definitions and standards for expedited reporting. Standard definition and terminology for key aspects of safety reporting, provide guidance for handling expedited reporting of AEs. E2C, clinical safety data management, PSUR, format and content of safety updates for regular authorities. And E2CA, there's other sort of addendum to E2. E2D being post-approval safety data management, the definitions and standards for expedited reporting. E2E being pharmacovigilance planning. And there's other ICHE guidance. And of course, FDA has the Drug Watch. All right, Drug Watch communicates emerging safety information while FDA continues to evaluate the drug. And Drug Safety Oversight Board will decide what drugs to be listed. And they're following up on the new or emerging safety information, which could affect prescribing decisions, if measures can be taken to prevent or mitigate harm, and whether an approved off-label use of drug poses significant harm. So these are a lot of thinking from the post-marketing point of view. All right, so let's start with exposure, all right? So first, the extent of exposure, dose, duration, number of patients. Like I said, the three dimensions, dose, duration, number of patients should be examined to determine the degree to which safety can be assessed from the study. The next thing we'll look at reasons for study discontinuation. Well, study discontinuation really affects the duration of exposure. So again, this is still part of the denominator. And you want to know not only at what time the patients discontinued, but also reasons, because if patient discontinued because of lack of efficacy, because of moving out of town or some other things, you know it's not related to safety. But if you look at the reasons in the test trial group, is mostly for safety. For the control group, it's mostly for non-safety. Well, that will give you some warning signal. All right. So patients who drop out of the study due to adverse events, loss of follow-up, poor compliance, patient decision, etc. Now we talk about AE. Again, I want to say AE is subjective. Is that the event that can be can be detected by the patient and can be observed by the physician. All right. So the more common adverse events should be identified, classified in some reasonable way, and compared for treatment groups, and analyzed as appropriate for factors that may affect the frequency of AE, such as time dependence, relation to demographic. So we typically don't do a whole lot of extra analysis like this for a single clinical trial. We tend to do this in the combined analysis. All right. Lab is objective safety findings. All right. Clinical chemistry, hematology, and many other things. Vital signs, ECGs, similar to lab, these are objective findings. SAE, all right, serious adverse events and other significant adverse events. Finally, serious adverse events and other significant adverse events should be identified usually by close examination of patients who left the study prematurely because of an AE, whether or not identified as drug-related. So these are the things we're going to cover today to look at sort of safety evaluation. All right. So SAE has a special definition. Only under this definition is considered SAE, serious adverse events. All right. So before I get into the SAE, I want to describe what a AE case report form look like. As we know, when we conduct clinical trials, we collect clinical data from the case report form. So at the design stage, we have to study design stage. We have to design case report form to collect the clinical data. So the AECRF case report form, based on my experience at Pfizer and my experience at Boehringer Ingelheim, they're pretty standard. So if you go out to most companies, I believe they're standard. So let me just describe what a AECRF look like. So on the CRF, of course, you have to collect drug name, well, study drug, right? Drug name, drug number, or whatever, and protocol number and center number, and then patient ID. So this patient identifier number, and visit number and visit date. All right. Now for this AE, so one page for each AE. Now for this AE, there's AE name. Well, it could be headache, could be nausea, vomiting, could be whatever, the AE name. And then severity, right? So there's 3 levels of severity. It's mild, moderate, or severe. So for every AE, it can only be classified in one of the 3 levels of severity, mild, moderate, or severe, right? And then start date, stop date. When did the AE start? When did it stop? All right. In clinical data management, I think the biggest challenge is that by the time you complete the study, you can find many AE case report form. There's missing AE stop date. This is because during the clinical visit, the patient reported the AE, and so there's a start date, and the physician says, "Did the AE resolve?" No, I still have that AE. And in the next visit, the physician forgot to ask, and the patient forgot about that AE. So you didn't have a stop date for that AE. And so in clinical data management, I think they always pay attention to the AE stop date. So all right, let's go back to the CRF. All right, AE name, severity, start date, stop date. And sometimes they have a question about relatedness. Do you think this AE is related to the study medication? And then there's action taken. All right. The action taken, there are, for most AEs, there's probably no action, and the other common action would be prescribe concomitant medication to control this AE. All right, so it's no action or prescribing concomitant medication. These are the most popular way of handling AEs. Now, in certain protocols, if the trial protocol design allows dose reduction, then for those trials, the physician may decide to reduce the dosage of study medication, so then you have dose reduction as action taken. In some other situations, the physician may decide to temporarily discontinue study medication, maybe for a week, for extended period of time or something like that. So these are the potential action taken. So the most, I think the most consequential action taken is permanent discontinuation. So these are the list of actions taken under the AE case report form. So typically, the study design is that you allow one AE for each page. So there, for each patient, there's going to be many pages of AE. And so for every AE, the clinical monitor will have to, excuse me, fill out a form associated with that. Now, the reason I want to bring up this CRF design is that directly relates to the understanding of serious AE. Serious AE is not severe AE. That's fundamentally different. Now, serious AE are the events like described here, results in death, life-threatening, hospitalization, disability, incapacity, or congenital anomaly. So whenever these happen, we have to expedite reporting to regulatory agencies on blinding. I mean, whenever there's SAE, we have to take immediate action. Most cases, SAE does not happen during the clinical visit, right? So death, life-threatening, hospitalization, it doesn't happen in the clinical office, all right? So SAE does not have a case report form, all right? So the way you handle SAE is totally separate from the way you handle AE, all right? So SAE, there you have to take a special action on these kind of stuff. And so typically SAE are stored in a separate database. And the routine AE are stored in the AE database. Now I want to describe that AE CRF because when you report AE, you have to base on the data you collect from AE CRF. So we're going to spend some time to look at how you report, how you tabulate kind of things for AE. SAE, just a special situation. All right, other significant AE, like marked hematological or other lab abnormalities, that depends on company. Different companies have different ways to handle it because most companies handle lab just as lab. They don't link with AE. All right. AEs led to intervention like withdrawal, dose reduction, concomitant medication. So these are the action taken part I was trying to describe. Okay, other significant AE some lab abnormalities or AEs lead to, but these are not really sort of very strong action. I think withdrawal is a very consequential action, but dose reduction, concomitant medications could be common depending on the protocol design. So, like I said, in the AE case report form, there's protocol number, patient ID, visit date, name of AE, severity, start date, stop date, related study drug, action taken. All right. So the AE report has to be based on the case report form. So based on what kind of data you collect, and then how do you report? And there are three kinds of displays. There's tables and graphs, listings, and narratives. All right. Now, let me see if we can take a look at the table here. All right. So this is a table I copied from the Lipitor label. All right. So this table here is that if you look at Lipitor, look at 10 milligram, 20 milligram, 40 milligram, 80 milligram, and placebo. So a typical AE table looks like this. Under each treatment group, there's a total sample size. And typically, we're going to have 100%. And then they're going to report for all the events, the observed events, N and percent. N is number of patients. It's not number of events. That's very important, all right? The reason that we report events, we report patients, not events, is because that sometimes one patient may have multiple events. So you only count that patient. You don't count that number of events. A simple example would be like this. If drug A causes 20 events from 2 patients, and drug B causes 5 events from 5 patients, which one is more sort of potentially toxic? Of course, it's drug B, because there are 5 patients having events. So drug A has 20 events from only 2 patients. So it's just those patients are not tolerating this drug, but most other patients, they can tolerate it. So we report number of patients. We do not report number of events. And then the percentages is percent of patients. So here, if you're looking at the 12.9%, that means out of 3908 patients, 12.9% of those patients are having nasopharyngitis. All right. Well, placebo is still rather high. You know, you got 7311 patients and 8.2%. Of course, this label is based on combined studies. You have all the Lipitor studies up to the time they're preparing this label. So you have such a large sample size. But typically for each clinical trial, you may have a few hundred patients, or could be 50 or 70 patients. And then you have N%. And then these AE names is going to follow this MedDrug dictionary. All right, so earlier I talked about, when we were talking about ICH, I think ICH made a lot of important contribution. Of course, the most apparent contributions are the guidelines. So the ICH-Q, ICH-S, ICH-E. Oh, by the way, there's also ICH-M, M being multidisciplinary guidance, which is across all the CMC, preclinical, and clinical. All right, so ICH guidance. That's important contribution. But also, they came up with this idea of CTD, Common Technical Document. Common Technical Document being, you know, in US, if you file, the company want to do a submission, you file NDA for a small molecule, which is based on NCE, new chemical entity. So you can file a NDA, new drug application. But if you're developing a NBE, new biological chemistry, a new biological entity, that will go to CBER. All right, so NCE go to CDER, Center for Drug Evaluation and Research. NBE goes to CBER, which is a Center for Biologics Evaluation and Research. And if you do a large molecule, they call it BLA, Biological License Application. If you go to Europe, they call it MAA, Market Authorization Application. And you go to Canada, they call it NDS, New Drug Submission. Different countries have different names. But then ICH says, why don't we call them CTD, Common Technical Document, so that if you can put together one document, you can submit across all countries. So ICH, again, International Council on Harmonization, they try to harmonize across the globe of all the regular agencies. So, so in addition to the guidelines, the guidance documents, they also come up with the creation of CTD. And one of the most important contributions that ICH did was MADRA. So all these terms are based on the MADRA dictionary. MADRA dictionary is MEDDRA, Medical Dictionary for Regulatory Activities, MADRA. So later we're going to talk about MADRA. So let's come back and look at tables and graphs. So a table or an AE table is pretty much like what I just showed you. But for a single clinical trial, that table looks like this, all right? The left-hand column are those adverse events, all right? The adverse events are classified based on MADRA. And in MADRA, there are different levels of classification. So there's SOC, which is system organ class. And then they have multiple levels under SOC. And then we typically report at the preferred term level, all right? So later when we get to MADRA, we can see what do I mean by SOC and PT, preferred term, all right? So the leftmost column being SOC and PT. And then the next column will be placebo, all right? So placebo, we're gonna have N%. And on the top of the table, you're gonna have the total number of patients and with 100%. And then for each preferred term, you're gonna report N being number of patients experience that AE in percent by dividing the number of patients with events by the total number of patients for that treatment group. And then the next thing will be the test drug group, and you can have all other groups. So these are different columns for the AE table. We prepare a clinical study report. Typically, you have multiple types of AE table. All right. So later we're going to talk about TESS, which is treatment emergent signs and symptoms. So you may have a test table, you may have an all causality table, you may have a drug-related table, or so you have all the different kind of tables. These are the standard way of presenting adverse events. Graphs are not very popular, all right, but in certain companies, they like to see graphs, or maybe for certain drugs, you want to use graphs to identify the experience of AE. So the first thing we'll talk about is tables. Sometimes you use graphs. Listings. Let me give you an example of listings. All right. Now, let me go back to the basics, the fundamentals. That in clinical trial, every piece of data we collect, we have to report. All right. So we're obligated to report to the regular agencies about every piece of safety data we collected. So the listings is basically every page of AE become a line on your listing report, which is your listing of the AEs. So the listing goes like this. You have treatment group, right? So if I start with placebo, so within placebo, I have patient number 1, patient number 4, patient number 6, or whatever patient. And within that patient, I'm going to have the first AE, the second AE, the third AE. So every AE is a line on the case report form, okay? No, on the listing, all right? So every page of case report form translate into a line. Now this is a row, all right, a row. Now if you look at the column will start with PID and visit number, visit date, and then they'll get into so PID, visit date, visit number, visit ID, visit date, and then they get into AE name and severity and relatedness, and then action taken. So for that entire line that described the data you collected from that AE form, that page of AE form, and then you go to the next line, go to the next line, go to the next line. So that's listings. So we're reporting all the raw data to regulatory agencies. That's how we collect AE. Narratives is interesting. Narratives, not every AE get narratives, all right? And in certain drugs, the clinical team may define certain AEs they want to have narratives. So, so usually the narratives are written by medical writers or physicians, all right? And then for the data team to provide data for them to write the narratives, we have to provide not only these AEs, but also we have to provide the demographic data and other data associated with the patient so that the medical writer can write narrative. So let me describe the narrative for you. If you identify certain kind of AEs, you want to write narratives. So for that, within that protocol, within that study, every such pre-identified AE, every AE, there's a narrative. All right, let me describe this narrative to you, all right? The narrative starts like this. Patient ID, such and such, is a 59-year-old white male. The patient received study medication starting what date? And then on what date? Patient reported this AE. And after how many days this AE was resolved? And the physician took the following action to handle this AE. So that's a narrative. So you sort of describe the patient's experience for this particular event. So these are the narratives. So not necessarily every AE gets narrative, but for those AE that requires narratives, the medical writer will prepare all these kind of displays. So these will all be part of the clinical study report. So that's an example of AE table. All right. Now, study population is important. All right. So if you look at if you look at the study report, all right, CSR. If you look at CSR, for every study report, the table 1 is always patient disposition. So we had to count the number of patients in the study. So that's why in clinical trials, the denominators are easy because we have all the data available with us. All right. So your first table is patient disposition. You're going to have the number of patients screened, number of patients enter the study, and number of patients complete the study, and then number of patients discontinued before completion for different reasons. All right. So safety population include all randomized patients who received at least 1 dose of study medication. So that's the if you look at data analysis, at our first lecture, we talked about the patient analysis set. So safety analysis, you're analyzing as treated. So this is a treated set. So that's the requirement of the treated set. I mean, it has to be randomized and has to receive at least 1 dose of study medication. So that's your treated set for safety analysis. And by the way, for the both safety and demographic tables, the analysis set are both based on treated set. Only efficacy is based on intention to treat or full analysis set. So we come back to extended exposure, number of patients, duration, and dose. Okay, so the extent of exposure to test drug or investigational product should be characterized according to the number of patients exposed, duration, and the dose. Okay, so that's your denominator. Okay. So of course you can come up with different ways of presenting the duration of treatment, depending on the length of study design, right? I mean, if you have a long-term trial, you can break down in different kind of time intervals. So number of patients being exposed for specific time periods. All right. Now, the longest clinical trial could be many, many years. Like if you're doing an oncology trial, your primary endpoint is overall survival. You probably have to follow 3, 4, 5 years or even longer. All right. Based on my experience, the shortest protocol is 8 hours. All right. Dental pain study, you only follow the patients for 8 hours. That's it. So, and then even though for 8 hours there's patient to rescue medication and they can consider being the last time of follow up and all that kind of stuff. So you still have different kind of breaking down of based on the time length for the protocol. So exposure, of course, when we look at mean or median duration, I don't think we have to spend much time on mean or median dose because this only applies to dose titration designs. Number of patients exposed to specific specified daily dose. So these are on titration designs. So if we design for fixed dose trials, parallel trials for fixed dose, it's not that complicated. It is expected that total number of patients treated, including short-term exposure. So these numbers are changing. So depending on the medical condition, depending on the type of the drug, the requirements could be different. So what is some AEs increase in frequency or severity with time occur only after treatment with a drug for 6 months? Now, I don't think I can. I will finish the whole set of slides today. I think we'll have another lecture to cover safety. All right, but I do want to introduce this idea about late developed adverse events. I think in drug development, I think one of the biggest tragedy is late developed AE. So let me describe the situation. All right, we know that phase 1, phase 2 trials are typically shorter term. All right, so phase 1 trials sometimes just you can do a single dose, or sometimes a week or 2 or something like that. Phase 2 you can do probably 2 week, 4 week. These are very popular, or sometimes you go to 3 months. Or the longest phase 2 I know is 6 months. Now, if we're working on NCE, new chemical compound, new chemical entity, this is a chemical compound that the chemists synthesize in the laboratory, and we know that that chemical structure have never existed on Earth throughout the entire evolution. I mean, Earth is about 4.6 billion years old, and before the lab synthesized this compound, it never existed. And so in the human evolution history, no matter tens of thousands of years or even longer, human body has never been exposed to that substance. So now the patient is exposed to this substance. So what happens is that your body is going to react to it. And once your body reacts to it, it became an adverse event. So if you look at the clinical trial reports, if you look at phase 2 reports, usually during the first week or 2, the AEs pop up. And when you follow for a little bit longer, 1 or 2 or 3 months later, well, this AE died down because your body just get used to it. So the AE will sort of does not last for a long time. All right. So now the drug looks good. You have good efficacy and the safety is acceptable. So the company decided to move into phase 3 development. Phase 3, long term, usually 1 or 2 years, sometimes longer. And again, you follow these patients. And at 9 months, there's a new AE pop up. You never saw this AE before. This is a very difficult problem for a company to handle. It is a very, very difficult problem. Why? Because based on your PK data, you would assume after the ADME, remember, absorption, distribution, metabolism, excretion, the drug should be out of your body. Now, if that's the case, any of the AEs you should have observed in the first few weeks. But at 9 months, there's a new AE pop up. You have never observed that before. Now, this AE could imply that the drug did not completely excrete it out of your body. Right? So the exposure is sort of beyond our understanding from phase 1, phase 2 trials. So that's the first thing. All right, you don't want to see that happen, but it happens. The next thing is that when a late AE develops, there's nothing we can do about. All right, you don't want to stop the trial because phase 3 is huge investment. You've already committed. The drug will go to FDA, right? Now, after 9 months, you see some AEs and you want to stop development? No, you don't, because maybe after a little while, the AE went away. It may not be an issue. You don't know. So you keep observing. And when you follow up, maybe when you complete the entire trial, there's many of these late developed AEs. Are you still to file an NDA? Do you think the drug is safe enough? Now, if you want to do that, you have to come up with a way to explain why this AE does not cause more risk. And so all of these issues makes the late developed AE a very difficult situation to handle. All right. So we really don't want to have the AE frequency or severity increase over time. Because you usually found this in phase 3, and that's too late. AE collection, of course, most AEs are unsolicited. Well, I had a headache. So severity, causality, well, causality is kind of hard to say. It's more subjective, all right? Start date, stop date, action taken. Now, SAE is a totally different issue. I mean, I said earlier, right? Solicited AE, did you have a headache? We typically don't do this, all right? So when this happens, what you're going to see is there are going to be a lot of over-reporting of AEs. So MADRA, all right, I mentioned that this is a very important contribution from ICH, all right? Medical Dictionary for Regulatory Activities, all right? Remember I said SOC, System Organ Class, right? SOC. Of course, MADRA is not a static document. MADRA is a live document. So we try to standardize all the medical terms into MADRA. So I think right now, we're probably more than 26 SOCs, okay? And there's high-level group terms, and there's high-level terms and preferred terms, lower-level terms, extra terms. So we report AE table at the PT level, preferred term. All right, let me describe to you in the actual terms. That's where you collect it from CRF, the case report form. Well, the patient may report headache, may report nausea, vomiting, may may report upper left shoulder pain or something like that. So the physician or the study nurse will have to record this on the case report form. All right. So whatever wording they put it there is the actual term. All right. Now we have to map this actual term into the lower level term. And this lower level term will be mapped into preferred term. And the preferred term will be mapped to higher level terms, to high level group terms, and finally up to SOC. Okay, so when we report a AE table, when you look at the AE table, the leftmost column are SOC, and under SOC, they're the preferred terms. All right. Now, so here's an example. All right. Now, the actual terms could be the patient reporting body aches. All right. So that mapped into generalized aching, mapped into pain and discomfort, and mapped into general system disorders, and mapped into general disorders and administration site condition. So when you report this AE, you report at the PT level pain. Okay, under this SOC. All right. Now another patient reported pain upper right side, right upper side intermittent. So you mapped into pain and you mapped it up. Or the patient reported body pain, it mapped up that way. Or another patient reported chest pain, that's the actual term that mapped into it. All right. So for the data management process, what happened is that before you report AE table, all these AE CRFs will go through this auto encoding program. So this computer program will interpret all the actual terms into the Medra dictionary terms. So the computer will do that. So after auto encoding, there's still many terms that cannot be mapped. And then a physician or a medical writer will have to manually map these terms into the Medra dictionary. So only after all the terms from all the CRFs of this protocol being completely mapped into the Medra, and then you can run this AE table. So I think I will stop here. And this is slide number 29, slide number 30. And next time we'll continue from here. Thank you.